Hello everyone, I'm Sagar Banushali and I am going to talk about Glycocalyx. Suppose if I take up a bacterial cell, then Glycocalyx is the substance that is secreted from inside of the cell to outside, which is further deposited on the cell wall. This glycocalyx is viscous and gelatinous polymer which is made up of polysaccharide and polypeptide. That is first property if it is, I can say is it is viscous. Second property, it is a gelatinous polymer which is made up of polysaccharide and polypeptide. Okay, so third, if the glycocalyx is firmly attached to the cell wall, it is known as capsule. When it is firmly attached to the cell wall, it is known as capsule. When it is not firmly attached, it is known as slime layer. This glycocalyx is also known as sugar coat, which is covered on the cell wall or to the exterior part of the cell. So it is also known as sugar coat. To determine the presence of glycocalyx, it is important to do negative staining. So I can say that if I want to determine the presence of glycocalyx, I have to perform negative staining. Negative staining. Okay, so this was all about glycocalyx. It is viscous gelatinous polymer made up of polysaccharide and polypeptide. If it is firmly attached, it is called capsule, and if it is not firmly attached, it is known as slime layer. It is also known as sugar coat. If I want to detect the presence of glycocalyx, I have to perform negative staining. So this was about the properties. If I want to talk about the functions, I can say that first one, glycocalyx contributes to the bacterial virulence. That is, bacterial contributes to bacterial virulence. Bacterial virulence means the capacity of a bacteria to cause a disease or the bacterial pathogenicity. For example, if I take an organism called Bacillus anthrax, which is the causative agent of anthrax disease. Bacillus anthrax, if I take the strain, and it's two types, one is capsulated, another does not have capsule. The organism with capsule causes anthrax, whereas the bacteria or the strain without capsule does not cause anthrax. It simply indicates that the capsule is responsible for the disease. So it contributes to the bacterial virulence as the glycocalyx is responsible for the disease. Another example I can say is the organism which was used for Griffith's transformation principle experiment, that is Klebsiella pneumonia. The Klebsiella pneumonia, it had smooth strain and rough strain. The rough strain was able to cause the disease because it had capsule, while as the rough strain was not able to cause the disease because it didn't have the capsule, or the absence of capsule does not led to the disease. Okay, so this was about the bacterial virulence. Also, the capsule of the Bacillus anthrax is made up of D-glutamic acid. D-glutamic acid. This D-glutamic acid is a type of D amino acid which is very unusually formed. Okay, so the second function is adherence. Adherence. For a bacteria to cause a disease, it is important for the bacteria to attach to the surface, to multiply, and or to colonize and cause the disease. So the glycocalyx allows the bacteria to adhere to the surface. For example, the Streptococcus mutans attaches to the tooth surface via glycocalyx. This glycocalyx allows the bacteria to attach and to cause the dental caries. Okay, so this was about adherence. Third one, it prevents dehydration. The viscous layer that is present on the surface cell wall does not allow the movement of water from inside to outside, thereby retaining the water content of the cell. So the glycocalyx prevents the dehydration. Fourth one, it protects the cell from phagocytosis. Phagocytosis is getting engulfed by another cell. For example, if I take up the pathogen that invade our bodies, this pathogens are further engulfed or phagocytized by macrophages. So presence of capsule does not allow the macrophage or another cell to engulf this bacterial cell. So protection from phagocytosis. Another fifth one. Um, in some cases, the bacterial glycocalyx, which is made up of polysaccharide or polypeptide, is serves as a source of nutrient. In in low energy states, the glycocalyx, which is made up of polysaccharide and polypeptide, is further broken down into simpler products. So the organism can utilize those simpler products in low energy states and can survive in states. So as a source of nutrition. The example for the source of nutrition is S mutans, Streptococcus mutans. So the functions are contribution to bacterial virulence, adherence, prevents dehydration, prevents phagocytosis, source of nutrition. Example is S mutans. So this was all about Glycocalyx. If you like my video, please like, subscribe, share. Thank you.